Now we're recording. Start, count 5 seconds and you're gonna start.
I'd like to thank the organizers for allowing me to talk to you today. What I want to do is talk to you about the urinary microbiota, or what we sometimes call the urobiome. First, I'm going to give you a short history of urinary tract research to explain why people thought that urine is not sterile. Then I'm going to describe some diagnostic tools that we use for detecting urinary bacteria, what microbes we detect in the bladder, how the microbiome is different between individuals with symptoms versus those without symptoms. I'm going to describe why the standard urine culture test used by clinical microbiologists is flawed. I'm going to try to answer the question, is a urinary tract infection always an infection, or rather an invasion, or could it be a dysbiosis? So, a little history of my own. I'm a microbial geneticist and physiologist. I've spent most of my career taking E. coli K12 apart, trying to figure out the circuit board inside. We want to know, how does a bacterium respond appropriately to external and internal stimuli? And then about 15 or 16 years ago, I walked up to a urogynecologist, Linda Brubaker, and I said, we have two words in common, E. coli. I study it. You want to kill it? And she said, let's talk. Somewhere along the line, during our conversations, she told me that some people think that the bladder is sterile. And I said, that's not possible. How could that be? Have you seen the plumbing? Well, I'm not a plumber, but she is. Urogynecologists, of course, are surgeons that fix female plumbing. So we decided that what we really had to do was to test the dogma that urine was sterile. And at the time, 16S ribosomal RNA sequencing was becoming cheap enough that groups with small amounts of cash could actually do the work. So that's what we did. First, I'm going to tell you why people thought that urine is not sterile. And then I'm going to show you how we determined that the bladder is not sterile. Okay, so here's the old dogma. The bladder is sterile. Bacteria are not involved in most urinary disorders. Urinary tract infections occur when a uropathogen crawls up the urethra into a sterile bladder, and it's mostly about E. coli. The new reality is that most bladders have a detectable resident microbiota, right? Communities of bacteria and other microbes like yeast, viruses, right, that live in their bladders. Many urinary disorders are likely the result of disruptions of that resident microbiota, okay? That is a dysbiosis. And believe me, it's way more than E. coli. So let's go back to 1860s. And Pasteur is doing his experiments to disprove spontaneous generation. You remember these experiments, right? You learned about it in high school probably, where he took a vial of some fluid, let's say in this case urine, and he put it in a sealed container, and he took another vial of urine and put it in an open container, and the open container got cloudy, and the sealed container did not get cloudy. Now, what Pasteur concluded was that there was no spontaneous generation. The problem is that his results were misinterpreted. And 20 years later, Roberts wrote a very important paper where he starts it out with the quote, fresh and healthy urine is perfectly free from bacteria or other minute organisms. And that conclusion is based on flawed logic, because it assumes that urinary bacteria grow in aerobic conditions, right? Ambient atmospheric conditions, right? You just put it in a normal incubator, and urinary bacteria, if they existed, would grow. Except we have found that most of the bacteria that exist in a urine sample taken from the bladder do not grow under ambient conditions. They're not aerobic. They like 5% CO2 or even anaerobic conditions. And so those bacteria couldn't grow if you just put it in an incubator in ambient conditions. Okay? All right, now we're going to jump ahead about 70 years, 80 years, and we're going to talk about Ed Cass. Now, Ed Cass was a brilliant man. He was an infectious disease doc at Harvard. And what he wanted to know is who was at risk for kidney infection, pyelonephritis. And he's the one who developed the standard urine culture protocol that most clinical microbiology labs use today. And it was hugely successful because it efficiently detects E. coli, which is the most common cause of pyelonephritis. Somewhere along the line, the test was moved for diagnosing bladder infections, cystitis. But the problem is there was no good evidence, not the kind of evidence Cass had for pyelonephritis, that it worked for cystitis. There are a number of problems. First of all, one problem is that the standard urine culture test, or traditional urine culture test, is designed to detect E. coli and a few other fast-growing, non-fastidious bacteria. The ones you know, E. coli, Klebsiella, Proteus, Pseudomonas. These are bacteria that can grow under a variety of different conditions, and they're rapid growers. Okay? And I'm going to show you later on that the standard urine culture test is not, that it's flawed. Okay, so here's why we hypothesized that the bladder was not sterile. First, we knew about the human microbiome. Okay, there's one bacterial cell for every human cell. We are simply walking, talking incubators. We can't live without them. They feed us, they protect us, they educate our immune cells, they provide us with energy, they give us vitamins, all sorts of things, right? And we also knew that some organs that had been considered sterile turned out not to be. And the best example I can give you are the lungs. Finally, we knew that most bacteria in the world don't grow under the standard urine culture conditions. So here are some diagnostic tools for detecting urinary bacteria. We're going to use several of them in the work that we've done over the past several years. So there are two basic culture-dependent methods. The standard urine culture test, that's the Clin Lab test, and then an enhanced culture test designed to grow most of the bacteria that can be sequenced out of culture, standard urine culture negative samples. So our particular version of an enhanced urine culture test is called EQUUC, or expanded quantitative urine culture. Then there are a couple of different culture independent or DNA-based methods that I'll describe. One is multiplex PCR, and the other is 16S ribosomal RNA gene sequencing. The standard culture, I've already told you, is the one that's currently used around the world in Clin Labs. They're very good at detecting E. coli and other fast-growing aerobic bacterial species, but it misses the majority of urobiome microbes. EQUUC, on the other hand, detects everything that SUC does, standard urine culture, or abbreviated SUC, right? It detects both uropathogens and commensals, right? And allows us an assignment to the species level. It is slower and a little bit more expensive than the standard urine culture test. And it's really good for research and when SUC is consistently negative in the presence of symptoms. So you have a patient who has symptoms, chronic symptoms, and every time they give a urine sample, it comes back negative. EQUUC would be a good option under those circumstances. Well, I'm going to talk more about this later. Multiplex PCR does not require growth, so it's rapid. It's only focused on a subset of suspected uropathogenic species. Currently, it doesn't detect commensals, which I think is really important. You need to know what the microbiome looks like so that you can push it to favor the good guys and to disfavor the bad ones, right? and it's good for rapid diagnosis. 16S ribosomal RNA sequencing is the most comprehensive of these tests. It detects almost all the bacterial taxa with outgrowth, and you can get species level assignment for some, but genus level for many. And that's because 16S ribosomal RNA sequencing requires that you get short reads using next generation sequencing platforms. And those short reads do not have enough information sometimes to distinguish closely related organisms. But it's not fast, it's expensive, it's mostly useful research. And I am aware that some companies offer it for diagnosis. I also think that's probably not a great idea because most clinicians have no idea what to do with the organisms that 16S ribosomal RNA sequencing detects. In fact, probably half, if not more, of those organisms, I don't know what to do with. I don't know whether they're good, bad, or indifferent. Okay, so now I want to talk about some of our findings. First, I'm going to show you some of the work that we did to show that there was DNA evidence of bacteria in standard urine culture negative samples that we obtained directly from the bladder. Okay, then I'm going to show you how we determined that the bacteria were actually alive, how we developed that enhanced culture technique, EQUAC. Then the question is what bacteria do you normally see in asymptomatic continent women? And then talk about what's different between individuals that have symptoms and don't have symptoms. And then a few other newer studies at the end. Okay, let's start with that first publication 10 years ago. So the first is how do we obtain urine from the bladder? That became very clear to us at the very beginning that the only way to know whether the bladder was sterile was to obtain urine from the bladder directly. And that's when I learned about transurethral catheterization and suprapubic aspiration. For those who don't know, a suprapubic aspirate is where you put a needle through the lower abdomen into the bladder and you withdraw urine that way. The controls for that are skin swabs where the needle goes in and a sham needle stick that goes into the abdomen but not into the bladder. Okay, we then took midstream voided urines and vaginal swabs. We sent the voided urine to the Clin lab for a standard urine culture test, a suck test, and all the results came back negative. So now the question was, can we detect bacteria in culture negative urines taken either by suprapubic aspirate or transurethral catheterization, and do they differ relative to midstream voided urine and vaginal swabs? And the answer was yes. Okay, we use 16S ribosomal RNA gene sequencing, which, like I told you before, allows detection of bacteria without growth, right? And what we found was that the aspirated microbiome looked very much like the catheterized microbiome. And too often, they did not look like the voided urine, which too often looked like the vaginal swab. Okay, so since the aspirate bypasses vulvovaginal contamination, I should have said these are all adult women going in for urogynecological surgery. Since the aspirate bypasses vulvovaginal contamination, the detected bacterial DNA must be from the bladder. I should say that the microbes that we saw in the aspirated sample did not look like the controls, the skin and sham needle stick. And the catheterized urine, since it looked like the aspirate, suffices for the study of bladder bacteria because it is considerably less invasive than suprapubic aspiration. We also determined that midstream voided urines were not a good way to determine what was going on in the bladder. Okay, and I'll show you a little more data about that later. So then the next step was to ask, well, geez, if we can see bacterial DNA in standard urine culture negative urines, can we grow those bacteria? At the time, Paul Schreckenberger was the director of our clinical microlab, and he was a big supporter, one of our first collaborators, and I urged him to develop a test, and he came up with EQUAC, and we had a master student by the name of Evan Hilt who established it. And this is the difference between standard urine culture and expanded quantitative urine culture. All right. So suck is 1 microliter of urine, put on a blood agar plate, a McConkey agar plate, put under aerobic or ambient conditions, and grown for about 24 hours at 35 or 37 degrees. For EQUAC, we use 100 times more volume, multiple different growth media, multiple different atmospheric conditions, and we let it go for twice as long, 48 hours. Here is one of the 2 standard urine culture conditions. 1 microliter put on a blood agar plate and incubated for 24 hours, and there's nothing on this plate. The clinical microbiologist would tell the clinician, no growth. Okay, what I'm going to show you in a moment is the same urine under one of the growth conditions for EQUAC. But remember, it's going to be 100 microliters, and we're going to incubate for 48 hours. So this was the same batch of blood agar plates, the same urine, but if you allow it to go for 48 hours, you start to see all these little colonies here. They're slow growers. They were not detectable after only 1 day of incubation. And these big colonies, there are not enough of them. You needed 100 times more urine to be able to see it. So this urine's not sterile, and this is a really good example of why suck sucks. Okay, I mean, in English, right, suck means it isn't very good. That's why we named it that. Okay, now that we know that the urobiome exists, we've detected bacterial DNA in standard urine culture negative urines. We've shown that we can grow them. They're alive. Who are they? So now I'm going to show you some work from another student of mine, Travis Price, who looked at the microbiota of our controls. And there were 224 continent controls, right? And this is all EQUAC. We've done the same work with 16S sequencing, and we get very similar results. Okay, so the black are negative, right? So they were EQUAC negative, but when you sequence them, about half of them, and we only sequence 1 mil, right? If you sequence 1 mil, about half of these that were negative turn out to be sequence positive. And if you sequence 20 mils, almost every one of them comes up positive. We like to sequence 1 mil because we can. It's more efficient. In any case, what you see in those that are positive is a predominance of lactobacillus, followed by Gardnerella, Streptococcus. Here's some staph. Remember, these are asymptomatic women. They're mostly perimenopausal, right? So they're older women. Here are people who are almost entirely colonized by E. coli, right? But they have no symptoms. And what you see here is that typically one bacterial genus dominates, and it's most often Lacto, Gardnerella, Streptococcus, and so on. Okay. What we also found is that Gardnerella seems to be a genus of younger women that tends to be replaced by Escherichia E. coli in postmenopausal women. So E. coli is common in older women. Gardnerella is found mostly in younger women. What about men? Well, we did a study on men with benign prostate hyperplasia. We took voided urines, catheterized urines, and what you can see is that the voided urines were typically positive. The cath urines, not so much. But even when, and that's because the voided urine is sampling the urethra, and there's more biomass, there are more bacteria in the urethra than there are in the bladder. You can see that in some cases, like here, the Streptococcus is present in the voided urine, and you see it in the catheter. Okay, you can see here's some staph that's in both. Here's Gardnerella in both. But you can clearly see that there's Candida in the cath sample over here, but not in the voided sample. And even when you see, let's say, the Streptococcus in both places, the voided urine is much more diverse. There are more other bugs. Okay. We also found that the tendency to be positive in the bladder was associated with increased symptoms, suggesting that the possibility that that's at least symptoms associated with benign prostate hyperplasia is associated with microbes. Okay, so let's talk about differences between symptomatic and asymptomatic subjects. All right, and this is another study by my former student, Travis Price. We're going to look at UTI versus non-UTI. So here's a pie chart looking at whether or not there's E. coli, other UTI-associated microbes. This would be Klebsiella, Proteus, Pseudomonas, microbes that are typically not associated with UTIs, like Lactobacillus or Gardnerella. Culture negative is black, and then polymicrobial circumstances here, right? And what you can clearly see by looking at the two pie charts is that women with UTIs and women with controls look very different. And it is true that UTI is an E. coli problem, right? About half of these women were predominated by E. coli. And another, I don't know, 20% were dominated by other typical UTI-associated microbes. But these people had symptoms. And these are microbes that are not usually thought to be associated with urinary tract symptoms. And here are polymicrobial infections. And in fact, microbial infections are considered to be, if there's 3 or more microbes on a plate, clinical microbiologists typically call that contamination. We find that that's not true. So here's the deal. Only about 50% of older women have E. coli associated with their UTI-like symptoms. The other 50% not. Furthermore, E. coli is common in older women. And in this case, this is greater than that threshold that the standard urine culture test usually uses, right? 10 to the 5 colony-forming units per mil. What Travis found in an earlier study is that thresholds are poorly associated with symptoms. If you see 10 to the 5 colony-forming units of E. coli, it does not mean that someone will have symptoms, okay? And that was true for other species as well. So the whole idea that 10 to the 5 or 10 to the 4 colony-forming units of some typical uropathogen does not hold up often enough to be used, in my opinion, as a reliable test. Okay, so I'm not going to tell you that that UTI is not an E. coli problem. It certainly is, right? But I'll show you now that standard urine culture, the test is flawed. So here's another study by another student of mine, Bailey Hochstetler, who's still in my lab. And what we're looking at here is the frequency of detection, right? How common is it? And these are a bunch of organisms, some whose names you probably recognize, and some you're going to go, what? Never heard of them, right? What we did is we took paired catheterized and voided urines from older women with recurrent UTIs, and the catheterized urine was assessed by EQUAC and by SUC, the voided urine by EQUAC and SUC. So the gray is testing by EQUAC, the black is testing by SUC, the solid is catheterized urine, and the hatched bars is the voided urine. Let me show you some. I'm going to highlight. These are the typical uropathogens: E. coli, Klebsiella, Proteus, Pseudomonas, and relatives. And what you'll notice is that SUC does a pretty good job of detecting E. coli, Klebsiella. So here, what we're talking about, here's catheterized urine, SUC, here's EQUAC. Okay, not much difference. Okay. EQUAC did a little bit better than SUC for Klebsiella. For the cath urine, pretty much the same for the voided urine. And then over here, they're pretty much identical. So these are the fast-growing facultative anaerobes, the bacteria that can grow under anaerobic conditions or aerobic conditions, ambient conditions. SUC is fine. But here are the organisms that SUC does not detect or detects very poorly. And they are mostly the organisms you've never heard of before or only vaguely. Actinotignum shallii, Aerococcus urinae, Alloscardovia omnicolens. Here's a yeast, Candida. Streptococcus agalactiae, well, that's group B strep. And here's Streptococcus aeruginosus, which is very often detected in women with urgency urinary incontinence. It's the most common species that we see. And now I want to point you to an organism that you're probably all familiar with, considered it to be a typical uropathogen, and I'm going to show you that suck can detect it, but it doesn't do it often enough. And that's Enterococcus faecalis. Look at this, right? Here's cath urines, and suck detected E. faecalis, but it missed it most of the time in the cath urine, and it did a very poor job of detecting in the voided urine. Okay, so that means that someone using the typical clin lab test would underestimate the presence of Enterococcus faecalis. So now, is a urinary tract infection always an infection? Or translate that into, is it always an invasion? Okay, so I want to ask you the question, how do bacteria reach the bladder? And for those who understand bacteria, at least E. coli and Proteus and the typical uropathogens, you go, well, they have propulsive devices. They have flagella, the propellers, right? They have flagella that they use to swim, and they have pili, that are retractable, so they're like grappling hooks, so they can crawl against the downward stream of urine and make their way through the urethra into the bladder, and in some cases into the kidney, right? But I want to propose to you a little bit of a problem, because most of the bacteria that we detect in the bladder are not modal. They have no propulsive devices. They don't crawl. They don't swim. I spent a good chunk of my early career studying bacterial motility, and I know what a propulsive device looks like, and I know what the genes that encode those propulsive devices look like. I can tell you these things are not modal. So how in the world did they get into the bladder? We're talking Lactobacillus, Gardnerella, Enterococcus faecalis. I have no idea how they get in, but they do get in. I guarantee you it takes a long time. So if you have a non-modal pathogen causing urinary tract-like infections, then I want to know how in the world the bacteria got there. Well, let's just talk about E. coli, right? So there are two possibilities. An E. coli UTI is always an invasion and therefore an infection. Or perhaps the E. coli got there a long time ago, and all you're seeing is a disruption of the microbiome, where the E. coli or whatever the other pathogen is, right, blooms. That disruption is called a dysbiosis. So most people think that UTI is caused by E. coli or other uropathogens, that those bacteria come from the gut. Well, okay, we're testing that right now. All right, we're recruiting women with UTI-like symptoms. We're taking urine by a transurethral catheterization. We're getting a rectal swab. Both of those samples are coming to my lab, and we're enriching for E. coli. We're letting them grow overnight so that we can grow up the E. coli. In the meantime, the cath sample goes to the clin lab for the typical standard urine culture test. And if that test comes back as positive, we then shotgun metagenomic sequence. We're going to sequence without amplification. We're not talking 16S, but we're sequencing everything. We get the human DNA, we get the bacterial DNA, we get yeast DNA, and so on and so forth. If we sequence deeply enough, then we can get strain level comparisons. So what we're asking is, do you see the same strain of E. coli in the bladder and the rectal samples? And if so, that's evidence that there was an invasion. Okay? You got the same strain of E. coli in both places. But what if we see one strain or a set of strains in the rectal sample and we don't see any of those strains in the bladder? That can't be an invasion. There's E. coli, UTI-like symptoms, but the strains that are in the bladder do not resemble any of the strains that are in the rectal sample. Well, that's got to be a dysbiosis. We see both scenarios. So these are early days, but we have at least one case where it's absolutely certain that the E. coli in the bladder is not from the gut. Okay, so This is not, we haven't published this yet. We're still working on it. We actually have enriched for all sorts of bacteria. So we're going to be able to report on more than E. coli. Okay. Lastly, now that we know the urinary microbiota exists, the question is, do those bacteria really live in the urine? Or is the urine a proxy for bacteria that are associated with the urothelial tissue? Look, if bacteria in the urine, there's two problems. First, urine is a lousy, really lousy growth medium for most organisms. Okay. And second of all, if in the urine, they're on their way out. So I've always thought that we were using urine as a proxy, that in fact, what we were seeing is the sloughed off bacteria from the urothelium where in fact they are colonized. So the question is, can we detect bacteria in urothelial biopsies? So we hooked up with Steve Walker and his team from Wake Forest. He provided bladder biopsies that were obtained from female women with PBS or painful bladder syndrome symptoms and asymptomatic women of the same age range. What I'm going to tell you is that we see no difference between the asymptomatic women and the affected patients. So this is not really about IC-PBS. I think that IC is not a microbial problem. I think it's maybe neurologic or something. It's outside of my range. It's not microbiology. But these biopsies allowed us to determine whether or not we could detect bacteria in the urothelial tissue. So we use 16S ribosomal sequencing. We use the V4 region. That's the portion of the 16S gene that we sequenced. And then we used imaging, fluorescence in situ hybridization, or FISH, in immunohistochemistry, or IHC. So first things first, wanted to make sure that the samples that we sequenced did not resemble the negative controls. So this is a way to show you that the samples are distinctly different from all our controls, okay? So that we're not accidentally studying contamination. We're very careful about that. And here's what we got when we were done. All right, we're looking at relative abundance, that is, what percentage of the total was light blue, that's Staphylococcus, or Escherichia, that's gold. And these are all of the samples here, and most of them we have duplicates, A's and B's. And what you see is that Staphylococcus is both prevalent, it's very common, and it's very abundant. Lactobacillus is common and abundant. Escherichia and Gardnerella are common, they're prevalent, but they don't seem to be abundant. Okay. Interestingly, what we are seeing is the same organisms that we see in urine, at a different ratio, and that makes sense because some organisms are going to be deeply embedded in the tissue and they're probably going to show up very rarely in the urine, whereas others are probably on the surface of the urothelium and they're probably sloughed off into the urine quite a bit. That's a hypothesis of mine. I don't have the data for that, but we will go after that problem. We will test that, my hypothesis. Okay, so now here's the imaging. This is just one slide, one biopsy, but cytokeratin shows you where the urothelial cells are. The 16S probe, there's a probe that's detecting 16S ribosomal RNA, and that's the red. The blue is DAPI staining, that's staining the nucleus of the urothelium, and then you merge them, and what you can see is that the bacteria seem to be surrounding the nuclei of the urothelial cells, at least in this particular biopsy. We have lots of other biopsies that give us similar results, and that paper is almost ready for submission. So let me conclude. The lower urinary tract is not sterile. The bladder is not sterile. And given that, if everybody is bacteriuric, what does significant bacteriuria or asymptomatic bacteriuria really mean? Maybe these are terms that need to be retired, or at least differently defined or more precisely defined. E. coli is common in older women with no symptoms, so the presence of E. coli does not necessarily mean that person has a UTI. This is a particular problem for non-responsive individuals. Okay, so Geriatric urologists have a lot of problems with this. I showed you that suck is inadequate. Your data may be flawed. In fact, in some cases, I guarantee it is. UTI is clearly more than just E. coli, because look at all those organisms that we saw in recurrent UTI patients. It was not just about E. coli or Klebsiella, but there was Enterococcus faecalis and all those organisms that you probably never heard of before. And yes, we can detect bacteria inside the bladder tissue. So now I want to challenge you to think differently. Maybe what your teacher taught you in medical school or in graduate school or wherever you went to school, maybe what your teacher taught you is no longer true, or maybe it never was true. And I want to thank the team that does this work. I'm just the figurehead here. There's a whole bunch of basic scientists. This is my group, present and past. Here are, we can't do this work without the analysts, the genomicists and bioinformaticians, the clinical microbiologists, and of course the clinicians. There are a lot of them. And then we have collaborators outside of Loyola, and we have funding. And with that, I'll thank you. And if you have any questions. We done?